This project we completed and published in 2018. And I'm sure a lot of you have heard the genomic side of the story. But today I'm going to focus on the transcriptome side of the story and show you some of the things that we used Iso-Seq to enrich our biological understanding of the great apes and the differences both genomically and transcriptomically between us and our closest relatives. So I like to put up this slide to just remind us that we are in fact apes, right? We're mammals, and specifically we're great apes. And so this is our family tree. The great apes, including gorilla, starting furthest out with orangutan, to chimpanzee and bonobo being our closer relatives, diverged from the other apes around, the lesser apes around 18 to 20 million years ago. And then most recently, it's estimated that we humans have branched off about 4 to 6 million years ago. And there really is an amazing amount of phenotypic variation within this family for such a short evolutionary time span. So for example, there's physiological differences, right? So humans, we're only bipedal, but gorillas, walk on their knuckles. There's dietary differences. And most interesting to my former postdoc advisor, Evan Eichler, right, is the cognitive differences and how the genetic architecture and differences between the apes predispose specifically human to genetic neurological diseases, such as ID, so intellectual disabilities, ADHD, schizophrenia, and other human diseases. And one of the most interesting differences is our brain size. And so chimpanzee, our neocortex, I think, is 3 times larger than the chimpanzee by volume, shown here in this diagram. So here is our human, and this is a chimpanzee. All right, so we started by sequencing the apes on the RS2 PacBio platform, and that was a while ago. And this was a massive undertaking at that time. We burnt through dozens and dozens and dozens of SMART cells. And the outcome is we generated high-quality, contiguous ape genomes. And this is the first time anyone had done long-read transcriptome sequencing of non-human primates. And we spent a lot of effort not only ensuring that the genome assembly turned out well, but we also made sure that the de novo genome annotation was done correctly and that we were able to trust the genes that we find. And so this picture kind of summarizes our efforts at the genomic level, where the picture here on the left represents a complete genome. And the picture on the far right represents the state-of-the-art genome assembly before we started the project. And for every contig, or sequence that is assembled, I moved a single pixel in each one of these pictures. And so here is the PacBio long-read genome assemblies. And basically, we're getting close to complete genome, complete contigs that are complete chromosome arms. And we scaffolded with Hi-C and BioNano to generate chromosome scale genome assemblies. Right? And so I like to show people these 2 figures because this is chromosome 3 of GRC38. And these are the 5 genome assemblies we did, 2 human, CHM13 being a hydatidiform, effectively haploid human genome. We did a Yoruban. Clint is the chimpanzee. G-Smart is gorilla. And Susie is our orangutan. And these blue blocks represent alignments. So these are Blazer alignments of contigs versus the human genome. And what you can see is most of the genome is represented in very syntenic alignment blocks between human and the great apes. Yoruban was a little bit less, the quality was a little bit less. But what I think you should be able to see is that as we go out through evolutionary distances, or we go down in this figure, what we will see is that the alignment blocks start to break up. And what that represents is divergence and large-scale structural rearrangements between the apes. And another way to visualize this is we can look at our assembly N50, and then we can look at our alignment N50 to GRCH38. So it's like the N50 of alignment on the x-axis, and it's your N50 on the y. And so here are all the not PacBio genomes down in this corner. Pantro 5 is a mix of technologies, but it was primarily short read based. And here are all the long read genome assemblies. And again, Susie PAB being orangutan is out here, not because the quality of the assembly is worse, but because alignment starts to get difficult at that phylogenetic distance. And so we generated lineage-specific variation. So what do I mean by that? We went through a process of comparative genomics, short-read genotyping, to determine which structural variances or which sequence differences fall into which lineage. And for humans specifically, we were able to find 20 megabase pairs of human-specific structural variation. And we generated a high-resolution map of where these structural variants happen across the genome. To be honest, there's not a real genomic correlation. These events seem to be spread out all across the genome. But I'm here to give an Iso-Seq talk, right? And so how do we use the Iso-Seq data? And to be honest, I didn't do the genome annotation. Ian Fittis did the genome annotation, and he used the Iso-Seq data for de novo transcript models. But my job was to try to identify biologically interesting candidates, right? Differences between the apes that might have a functional impact. And so Jason Underwood and I came up with this way to try to find transcripts that map differentially between human and the other apes. And so what you do is you take chimpanzee Iso-Seq data, FLNC, so full-length, non-chimeric Iso-Seq molecules, and you map it to the chimpanzee de novo assembly, and you map it to the human de novo assembly. And then for the human Iso-Seq, you map it to human, and then you map it back to chimpanzee. And this cute little baby is one of Jason Underwood's twins, but I can never tell which one's which. Right. And so, so basically, we map these reads. We get a cigar string, for those of you who know, and we count the number of bases for each transcript that map to human or chimpanzee. And we look for discrepancies. And this is only possible with accurate long reads. And so Iso-Seq was like the initial hi-fi or CCS, right? Because we did consensus on the Iso-Seq molecules. Right? And so I thought, isocross might be, Jason came up with the name isocross. It seems like a X Games, right? So we're cross-mapping. It seemed like a good name for an extreme sport, but yeah. Sorry. Sometimes I'm not very good at telling jokes, apparently. But so let's get back to science. So what you can expect for most transcripts is that they should map nicely along the diagonal. Human and chimpanzee are less than 2% divergent, so most transcripts should map equally well. And this is chimpanzee data that's mapped to human and mapped to chimpanzee itself. And so that's why there's this bias, there's this shift towards chimpanzee, because it's chimpanzee Iso-Seq. And then also we looked at the percent repeat of the transcript, the genomic region, so how much repeats are in there. And we found that repeats can really drive this signal, right? And so most transcripts map really well, but what we want to focus on are the transcripts that don't map well. And so this shows the same plot, but highlighted are a few genes here. And so we cluster this signal, right? We cluster this off-diagonal signal into genomic regions and ask, is there a co-localization of transcripts that don't map well? And what we can find are, I've just highlighted two genes, CARD8 and FADS. And so if we look at the CARD8 off-diagonal signal, what we have here is a figure showing the ref-seq genes up here. So these are the human. This is the human genome up here, and the human Iso-Seq. And these are the two forms, the two exons, the two types of transcripts that we're getting from card 8 in human. This is actually just Iso-Seq data. It looks like a genome annotation, but it's just really the Iso-Seq data itself. And then this plot down here shows you the synteny, or the similarity between the orangutan back. These are validated through backs. So this is orangutan versus human. And what you can see is the orangutan, and actually all non-human primates, have a 66 kb deletion. And if we look on that back and we map the non-human primate Iso-Seq data, what we can see is a 13 exon deletion that is specifically only found in human. And card 8 is an immune gene, so if anyone's interested in that, you can talk to me more. All these, like if you want to know more about any of these examples, you can come find me later. Okay. In the second example, we have a similar thing. And I was really excited to see this human-specific deletion because it's in a region that's come up for GWAS for human diet. So Rasmus Nielsen looked at Inuit populations and looked at differences, to try to understand genetic differences between the Inuit populations. And what they found is that this region seemed to be associated with diet. And it's a very complicated region. And a lot of credit goes to Max Doltry, a grad student in the lab, who figured out what was actually going on. Because I looked at the Iso-Seq data, and it's a very complicated event. And so it is a 60 kb intronic deletion. And so Evan's like, why are you showing me intronic deletions? But I was like, that looks cool. And then what Max found out is that the intronic deletion is actually drawing a promoter for FADS 2 closer to the promoter of FADS 1, and it's changing the expression of the abundance of the different isoforms. And so without the Iso-Seq data, the end of the story was we found an intronic deletion, end of story. But with the Iso-Seq, we were kind of able to identify how this non-coding variant could have a phenotypic effect, potentially. All right. And then in this last example, I'll show you. So that here is another gene. This is a gene, that CDC25C, that's involved in cell regulatory cycle. And one of the hypotheses about why our brains are bigger is that we have slower cell cycle in our brain, and then therefore we can grow larger brains. That's just one of the many hypotheses. And what we found was an insertion. And how we found that is if we looked at human Iso-Seq data, it maps really nicely to human, no surprise. But if we looked at the non-human primate Iso-Seq data, so chimpanzee and gorilla, we see these red dots here. And what these mean is that the Iso-Seq data doesn't map well through that exon. It's a forced alignment. There's lots of mismatches and shattering and sputtering of the alignment. But when we look on the chimpanzee, we see a couple things. So we see a novel gene annotation from cat. And then we see these nice in-frame exons in chimpanzee and gorilla. So we identified a human-specific deletion of an exonic sequence. Right, so the PacBio data really enabled us to one, build high-quality reference genomes, and two, the Iso-Seq data was absolutely central to us discovering really interesting biological candidates, because no story is really complete with just a genome. And so the synteny between these two data types is super powerful. Since I did this project, a lot has changed. Iso-Seq has come such a long way, both bioinformatically and library prep. So think about this. So we sequenced, I think, well over 100 SMRT cells for only the Iso-Seq data. Today, a single SMRT cell would do that whole project. And that to me is like mind-blowing. And over the course of 2 years, this whole project, many, many hours. Katie Munson is the UW PacBio sequencing director. And she spent weeks upon weeks in technician sequencing, and now you can just do it with a single SMRT cell. For human, for example, a single SMRT cell will get you, on average, 4 million transcripts. And there's a kit that's been put out by PacBio. It's the Express kit, and it enables you to do the library prep within 1 day. And so within 3 days, you can go from RNA to sequencing, through sequencing, and then to analysis, through analysis of data. And so there's been a lot of bioinformatic improvements in terms of the speed of the Iso-Seq pipeline, and so now it's basically turnkey. And luckily, if you care to learn more, we have Liz is here. Liz, you want to wave just so everyone sees? Liz is the Iso-Seq expert, so if you have any questions, feel free to talk with her. And with that, I'd be happy to take any questions.